Welcome to Signal Ahead. I'm Alfred. Today's briefing, Neuroscience, Computational, Saturday, June 20. This is the synthesis above the noise, not the headlines, but the deep currents beneath them. Let's get into it. Let me begin with a puzzle that, once you see it, reorganizes how you read nearly every headline coming out of computational neuroscience this year. Suppose I told you that a frozen artificial network trained on photographs or audio clips or the sprawl of internet text, and never once shown a brain, could serve as the common reference point against which two different human brains are aligned, or against which the spiking of a single neuron in a mouse's visual cortex is translated into a sentence about what the mouse is watching. You might assume the network had to be tuned, coaxed, taught something about neural biology. It wasn't. It was held fixed, like a ruler, and the brain data was bent to fit it. That is the non-obvious thread I want to pull on, because it runs through work that, on the surface, looks utterly unrelated. There is research mapping functional MRI activity onto the embedding spaces of vision, language, and audio foundation models, and finding that the trick is not building ever more elaborate decoders, but achieving alignment, that a relatively simple contrastive matching between brain and model embeddings does the heavy lifting. There is the rather astonishing project of decoding spikes from thousands of mouse neurons into free-form natural language narration, accomplished by mapping spike trains into the patch embedding space of a frozen vision language model, with no training on the language side at all. There is cross-subject alignment achieved without any shared stimulus between subjects, simply by anchoring each person's neural activity to the scaffold of a pretrained artificial network. And there is careful comparison of human and large language model accounts of shared semantic representation, finding selective convergence, the machine and the brain agreeing on some dimensions of meaning and not others. Read these one at a time, as the news tends to, and you get a string of clever gadgets. Read them together, and a thesis emerges. The artificial network has quietly become the coordinate system of the field. The shared latent space, that high-dimensional arena where concepts are directions and meaning lives in the angles between vectors, is increasingly supplied not by neuroscience, but by machine learning, and neuroscience is moving in to take measurements against it. Now, why should that be the organizing insight of where the domain stands? Because it represents a genuine inversion of the old project. For decades, the ambition was to read meaning out of the brain on the brain's own terms, to find the parameterization, the tuning curve, the receptive field that explained why a neuron fires. The honest admission embedded in this new wave is that those responses resist intuitive parameterization. We could not write down what a higher visual cortex neuron encodes. So instead of decoding the brain into our concepts, we are borrowing a machine's concepts and asking how much of the brain projects cleanly onto them. The success is real and the humility is real, and they are the same fact wearing two faces. But I want to be careful, because a coordinate system is not an explanation. A ruler measures, it does not tell you why the thing is the length it is. And this is precisely where the field's other half, its older, more mechanistic half, is pushing back, and the tension between the two is, I think, the real state of the discipline. Consider the work running a complete fruit fly larva connectome as a fixed dynamical operator, fitting no single neuron parameters, asking how much of a circuit's behavior the wiring diagram alone determines versus the physiological detail it omits. Consider hybrid biophysical models embedding learnable differential equations inside conductance-based neuron models, specifically to recover interpretable gating variables, unmodeled ion channel kinetics, rather than to hide them in a black box. Consider a decade of refinement of a stochastic resonance theory of tinnitus, which reframes spontaneous neural noise not as malfunction, but as an adaptive upregulation to restore information transmission near a sensory threshold. These are not coordinate system projects, they are mechanism projects. They insist on knowing why. So here is the argument I want to develop across what follows. The field stands at a fork it has not fully acknowledged. On one path, the alignment program powerful, scalable, embarrassingly effective, and quietly dependent on artifacts whose own internal organization we do not understand. On the other, the mechanistic program slower, interpretable, anchored in conductances and connectomes and noise, asking the why question the alignment program brackets. And the most interesting work, I'll argue, is happening where these two pressures meet, where a self-organizing network is shown to grow attractor connectivity and anticipatory dynamics from Hebbian plasticity and adaptation alone, deriving structure rather than borrowing it. In the next stretch, I want to take the alignment program seriously on its own terms, to ask what it actually buys us and what it quietly costs. Then we'll turn to whether the mechanists are right that a ruler without a reason is a measurement we cannot yet trust. Stay with me, the convergence is more fragile and more revealing than the headlines let on. There's a temptation, when you survey a field as it actually moves paper by paper, week by week, to see nothing but a scatter of unrelated preoccupations. One group is mapping the complete wiring of a fly larva's brain. Another is teaching a language model to narrate what a mouse is looking at, reading the spikes of individual neurons. A third is fitting potassium currents into hippocampal cells, while yet another studies the strange anti-correlation of heart and brain in the final hours of a human life. It looks like noise, but if you stand back and let your eyes soften the way one does before a pointillist canvas, a single deep current begins to show itself. And I want to spend this stretch of our conversation tracing it. The current is this: the field is quietly abandoning the dream of the single sufficient description. For most of the 20th century, the implicit hope of neuroscience was that there existed, somewhere, a correct level at which the brain could be explained. Find the right variable, the firing rate, the connection, the channel kinetics, and the rest would follow as a kind of bookkeeping. What the present moment is teaching us, again and again across these otherwise unrelated papers, is that no single level is ever quite sufficient, and that the interesting science lives precisely in the gap between levels. Consider the connectome work. Here is the most complete description of a brain we have ever possessed, every synapse of an entire insect, reconstructed from electron microscopy. And the question the researchers ask is not triumphant, but humble. How far does this perfect map actually fix the activity? They run the wiring as a fixed operator, fit no single neuron parameters, and compare it against randomized networks that preserve only coarser features. What they are really asking is how much the structure determines, and how much escapes into physiological detail the map never recorded. That is the gap made into an experiment. The complete diagram, it turns out, constrains the overall dynamical regime, how strongly, how richly a circuit responds, without pinning down everything. Structure speaks, but it does not have the last word. You hear the same theme, transposed, in the work on infant brain development, where structural modules and functional modules refuse to line up neatly across subjects. Rather than forcing them into agreement, the response is to make the assignment itself probabilistic, to say that a brain region has a chance of belonging to a subnetwork, and to measure the consistency between structure and function as a quantity that varies, rather than a fact that holds. Again, two descriptions, neither sufficient alone, and the science is the disciplined measurement of their disagreement. Now watch the current carry into a completely different room. The work on decoding the brain by alignment rather than complexity makes an almost philosophical claim that what matters in reading neural activity is not the elaborateness of your model, but whether its geometry lines up with the brain's. Concepts as directions in a high-dimensional space, meaning carried by relative angles. And the practical discovery is that a contrastive objective, anchoring neural signals to the embedding space of a foundation model, captures shared structure without needing to reconstruct every nonlinear detail. The fMRI signal averages over millions of neurons. The microscale computation is wildly nonlinear, and yet at the level where the averaging happens, a linear-ish alignment suffices. Once more, the right description is not the most complete one. It is the one pitched at the level where the regularity actually lives. This is why the cross-subject alignment work, anchoring different people's brains to a common scaffold provided by an artificial network, feels of a piece with all the rest. It gives up the old requirement that subjects must see the same stimuli to be compared. Instead, it borrows an external structure and asks each brain to find its place against it. The shared computational principle is not extracted from any one brain in isolation. It emerges only in the relation between brains, mediated by a third thing. And even the tinnitus work, a decade of reframing phantom sound not as malfunction, but as the side effect of a nervous system adaptively raising its own internal noise to keep transmitting information near threshold. Even this belongs to the current, because it refuses the single-level story in which more spontaneous activity simply means something is broken. The hyperactivity is maladaptive at one level and optimal at another, and the truth is the tension between them. So the deep current is not a new technique or a new organ. It is a change in epistemic posture. The field is learning to hold several incomplete descriptions at once: wiring and physiology, structure and function, neural geometry and artificial geometry, noise and signal, and to treat their mismatches not as errors to be eliminated, but as the very texture of what a brain is. The map is never the territory. But increasingly, neuroscience is willing to study the difference between them as the most informative thing it has. There's a temptation, when you survey a season's worth of work like this, to file each paper neatly into its own drawer, one about tinnitus, one about infant brains, one about fruit fly wiring, one about whether a simulated mind could be conscious, and to treat them as scattered points of light with nothing much between them. I want to resist that temptation because I think if you stand back far enough, the constellation does have a shape, and the shape tells you something about where this field is heading and what you ought to keep an eye on. Here is the through line as I see it. Across nearly all of this work runs a single bet that the right way to understand neural activity is not to describe it, but to align it, to find a shared space, an external scaffold, a foreign coordinate system, and then map the brain onto it. Consider the decoding work that maps brain measurements onto the embedding spaces of large foundation models. The striking claim there is not that this works, but why it works, that success comes from alignment rather than complexity. You don't need an elaborate, biologically faithful model of neural computation. You need a good shared geometry and a contrastive objective to land things in the right place. The same instinct animates the effort to narrate, in plain language, what individual neurons in the visual cortex are doing, by routing their spike trains through the patch embedding space of a vision-language model. And it animates the cross-subject alignment work that anchors different people's neural activity to a common scaffold borrowed from an artificial network, dispensing with the old requirement that everyone view the same stimulus. Notice what has happened here. The reference frame for understanding the brain is increasingly not another brain, and not first principles of biophysics. It is a foundation model. The artificial network has quietly become the ruler against which the biological one is measured. And there is a finding lurking in this corpus that should make us pause: the result that large language models selectively converge with the semantic representations humans share between their brains. Selectively, not everywhere, but along certain dimensions. That word is doing enormous work, and it is the first thing I'd ask you to watch, because the second-order implication is uncomfortable. If our most powerful decoders succeed by aligning brains to artificial embedding spaces, then what they reveal is the intersection, the overlap between how the model carves up the world and how the brain does. And an intersection, by its nature, is silent about everything outside it. The dimensions where brain and model diverge are precisely the dimensions these methods cannot see, because the method's whole logic is to find the shared scaffold and discard the residue. We may be building instruments that are exquisitely sensitive to the parts of cognition that machines already capture and structurally blind to the parts they don't. That is a subtle form of streetlight problem, and it scales beautifully, which makes it dangerous. Set against this alignment-driven wing of the field is a quieter, older commitment to mechanism, and the tension between them is, I think, the real story. The work that runs a complete fruit fly connectome as a fixed dynamical operator, fitting no single neuron parameters, is asking a genuinely deep question: how much of a circuit's behavior is fixed by its wiring alone, before you add a single drop of physiological detail? The hybrid neuron models that embed learnable differential equations inside conductance-based biophysics are trying to discover unmodeled currents while preserving interpretability. The continuous attractor networks that grow their own connectivity from Hebbian plasticity rather than having it pre-wired. These are attempts to keep the causal, mechanistic story intact, to insist that understanding means knowing how, not merely mapping onto. And here is where the philosophy paper earns its place in the season. The argument from intrinsic computational functionalism that whether a system is conscious depends on the computational structure it physically realizes through its own causal organization, not on any description we impose from outside, is at bottom a demand that we take the mechanism seriously and not be satisfied with input-output alignment. It is the same fault line raised to its highest stakes. Does the shared geometry constitute the thing or merely correlate with it? Simulated water, the old objection runs, is not wet. So what should you watch? Watch whether the alignment methods start being mistaken for explanations, whether my decoder lands brain activity in the model's space begins to be reported as I understand what the brain is doing. Watch the infrastructure, the great open archives of neural recordings, because whoever sets the format sets the questions. And watch, above all, for work that closes the loop, that takes an alignment-derived insight and validates it against a mechanism, against a perturbation, against the firing of an actual cell. That is where the two wings of this field will either reconcile or come apart. The brain is not obliged to share our scaffolds. The interesting science begins exactly where it refuses to. That's the signal for today. The noise will keep coming. We'll keep finding what's underneath it. This has been Signal Ahead with Alfred. Until next time.